First time I ever spoke in this room was actually at a Linnaean Society meeting, and it was with a bunch of paleontologists, talking about molecular systematics of fish and echinoderms. And soon after that, I was lucky enough to be hosted by David at the Natural History Museum as a welcome fellow, and there's a few other people here responsible for that, and many of you responsible for the work that I've been doing on planet helmet systematics over the years. Russ chose this title because I was too lazy enough to give him a title, and I would have been quite happy with paleontology meets parasitology. I wasn't quite sure what was going on in Russ's mind when he did the subtitle of The Changing Perspective of Fossil Parasites to Parasite Futures. And all I can think of is he was thinking about the people who would be in here, the age, the demography, and I can assure you that in spite of the demography that is in here, the usual demography of the Linnaean Society meeting means that the average age is about 85, and Liz Rollinson has to keep the defibrillator going. Right, so let's remind you where you are. We started with a little introduction that you're at the Linnaean Society. This is Carl von Linné with his lovely wig. After some years, of course, he started as a young man as a keen botanist. And you'll see the problems with the images in a moment. The image there is Systema Naturae, which is the first big treatment of the natural world. Linnaeus gave us the binomial naming system, and a quote from one of his botanical books is that if you don't know the name of things, the knowledge of them is lost. So fundamentally that's what I do. I'm about systematics and taxonomy, and reminded that the Natural History Museum has a few helminths in jars going back a few years. And I think most of us understand the species that we're working on, unless they're hybridizing. Many of us have a fair idea of the number of taxa in a genus that we might be working on, but I think some of us would struggle to really fully appreciate the diversity that would be in a family of parasites that you're working on, or the class, or the order, or the phylum. And as you go up that sort of animal kingdom set of criteria, still fewer of us would understand that diversity when we start thinking about hosts. Paleontology meets parasitology. Nobody funds this work, so this is more a sort of fun for me. And the first thing I need to check is, are there any paleontologists in the room? Excellent, that's a perfect audience for me because I can say anything and get away with it. Now, paleoparasitology, when a lot of us look that term up, actually find that it's been taken up by archaeologists and anthropologists. It's really largely dominated, using that term paleoparasitology, by those two groups, and concentrating predominantly on human diseases and diseases of livestock and so on. So this is the Richard III grave, that was pulled up some years ago, and Piers Mitchell, an anthropologist at Cambridge, sifted through the sacred soil samples and found Ascaris eggs and various other lovely things. So I'm not talking about paleoparasitology in this sense, but I just want to give you a quick overview as to how successful that area of research is. Here's another nice study. This is Viking Age material, really good quality eggs pulled up. Peter Nason was involved with this, and doing some basic PCRs and actually finding haplotypes that confirmed identities and particular population markers of these samples, all the way back from the Viking Age. And Piers Mitchell has got a very nice summary for those of you who are interested in human and livestock eggs in a journal of paleopathology. So I'm sort of leaving it there on that side from a paleoparasitological front, from an archaeological view. Just to say that Andy Briscoe is in the audience with me. We're looking at paleoparasitological side of things on this side, largely because we're interested in seeing whether or not we can push the limits of environmental DNA and to see if we can identify eggs of tapeworms. So at the top there are Dithylobothrium species and Taenia species. These are from relatively recent grave sites from the Nenets people in northern Siberia, and we've been doing sort of crude shotgun sequencing of the sacral area of the soil samples. These eggs were found by our colleague Sergei Slepchenko, and he's particularly interested in actually identifying the species of Taenia and Dithylobothrium, which we think we can do because we've sequenced all of these taxa very extensively for the entire group. There's lots of nice anthropological stories that they don't think the Taenia could possibly be from beef or pork and think it's from reindeer, because at the time this particular group, these are nomadic people, were not allowed to have and we're not keeping them, we're allowed to keep them. So that's as far as we're involved with that, and there's a sort of forensic side as well, as we're discovering maggots and so on in the same samples. So I want to take you a little bit further into the tree of life and the origin of metazoa, and to introduce you to the ideas of deeper time and why fossils might be interesting when we're trying to understand parasitology in a broader sense. Where did the parasites come from and how did that diversity grow? So that obscure tree on the right is a phylogenomic tree in this era of genomes. If we can pull together multi-gene data sets and we can estimate phylogenies. There's still huge problems with those data sets. Getting the right models, getting the right taxonomic sampling. It's highly problematic. And yet you'll see sort of error bars in there which demonstrate ranges of diversification dates. Now those are all based on Bayesian modeling systems, and where possible there are good fossil data which are calibrating the tree. But the soft-bodied organisms, things like many of the helminths that we're going to look at, frequently don't have, or simply don't have, fossil evidence. So where might we look? What might there be out there? Was a sort of a raison d'etre for a colleague of mine, myself, Kenneth de Betz. He's a paleontologist, and he and I were interested in reviewing the state of fossil parasites over the last 25 years since Simon Conway Morris did a review. So this got us into looking at patterns and processes, me from a parasitological perspective and Kenneth from a paleontological perspective. So if we take a tree for a moment and look at the left and see a phylogeny, and then look at on the right the name of those phyla with the numbers that you're seeing associated with those are the number of lineages that have independently become parasitic. So we don't perhaps realise that when you take a tree of life perspective, parasitism, that singular event which is generally irreversible unless you're a nematode-eating plant and you can sort of go in and out, happened at least 223 times. That's an enormously successful lifestyle. And of course, there's opportunities to understand convergence, the response of parasites to hosts, and so on and so forth. The other thing is we've been focusing on helminths, and I'm going to do my best not to mention schisto, but I might at some point. And only 20% of those independently derived parasites actually are from helminths. Many of the parasites, the breadth of the width of those blocks basically show the number of taxa that are involved. So the arthropods, it's the insect lineages, and some of the crustaceans, but predominantly the insect lineages which have become parasitic. Think of parasitoids and so on. So if we are truly trying to understand the evolution of parasitism and trying to understand parasitology in a broader sense, we clearly need to look way beyond just the helminths. And that might be useful when we're thinking about the fossil side. Now, my own interest, so what I'd hoped Russ would have asked me to talk about was a project that I've been part of for the last few years, which is looking at cestode diversity. I've been lucky enough to be part of a team funded by the NSF, looking at tapeworm diversity. We've got this great title from Janine Kyron, Tapeworms from the Vertebrate Bowels of the Earth. And what we've been doing in that project is to visit over 54 countries and to sample extensively the major vertebrate groups in which tapeworms are adults. So over 250 individuals involved in this collecting. I've got to point out, it might be because I'm severely red-green colourblind, but we never went anywhere. The London group stayed where we were, and our job was to do the sequencing of the various taxa that were coming in to be able to generate a phylogeny, which is work in progress. But I wanted to use this example of trying to understand how one might document diversity and how one might understand better parasite diversity. So if we look at cestodes, and from this particular project, on the bottom left of the screen are the number of vertebrates that we know to be in particular groups. So the most speciose group of vertebrates are bony fishes, over 32,000, and then birds, snakes, lizards, amphibians, and so forth. And then what we've got listed at the top are the names of predominantly cestode orders and some families for cyclophilidians, which by far are the most diverse. So if you want to orient yourself from parasites you may know and love, Difflobothrium will be the Difflobothriidae. Taenia will be hidden well into the side of the Taenidae inside the Cyclophilidia. In fact, hidden in there. So these are highly derived taxa that we're normally dealing with, and this is the broad diversity of parasites that are there. So if we're looking at vertebrate diversity and seeing how the parasites are spread, that's one way of looking at it. The other way is to do a sort of relative cesto diversity. In other words, asking in which of those groups would you find the greatest diversity of cestodes. In this case, cyclophilidians, as I said, are the most diverse, and those are clearly in the more terrestrial organisms, the birds and the mammals. Now, what do you do with that kind of information? So this is a work in progress for us, to try and understand the interrelationships of that particular group, and then to try and start telling some stories as to how that diversity came about. One way would be to take a phylogeny and to start mapping, for example, hosts that are used. Now, cestodes are trophically transmitted, they're complex life cycles, but relatively simple compared to many other helminths, and one could start trying to understand what was happening at those ancestral nodes, what was happening at major changes, major transitions, whether they're biogeographic changes, whether they're morphological changes, and so on and so forth. And you start getting almost immediately, because we have no dates and we've got very little information on this in the world of just so stories. Just occasionally that changes when we have really good groups like polystones, which are monogeneans. They have a simple life cycle, a direct life cycle, and they are often quite host-specific, and in fact we find them also quite site-specific. So when looking at a phylogeny of polystones, this is Olivier Bernot's work with others, it's actually quite compelling to take the hosts and take the evolutionary history of the hosts and plot on that a sort of an expectation that the parasites follow the hosts. Now, in parasitology, that co-phylogeny is quite rare. Lice and birds, very good. Monogeneans, pretty good, but it's not often the case elsewhere. Work from Olivier's group and others was also used even further by looking at continental drift in these particular groups and looking at the distribution of modern parasites of amphibians, and using the breakup of Gondwana to estimate what happened when. And this is a terrible rendition of a slide, and I apologise, Richard. This is some work from Richard Tinsley, who published this last year. Now, this is an important one because it relates to getting your data out after a long time. This is work that started in Richard's PhD in the 1970s, and not only did he wait till he was retired to finish this, but he made sure he had a grown-up son who was also a trained evolutionary biologist. So congratulations, Richard, on that one. But this is a system that is ripe for looking at dates and looking at patterns and processes of evolutionary biology. But it's a rare system, and it's one that clearly kept Richard going and still does keep Richard going through his work. So, taking a phylogeny and asking what can you do with this, asking about ancestral conditions, predicting host shifts, looking at major radiations, invoking biogeographic patterns. There are many things one can do, but what's missing? It's a just so story. Do we have direct evidence of what those relationships might have been through geological time? Can we put timing events on there, calibration events, so that we can begin to look at rates of change through time? And can we try and avoid circularity by relying on the host to tell us the story about the parasite, but actually get empirical evidence from the parasite to move us forward? So if we take that slide from the beginning, the work by Ahmed Kuris and Weinstein, and plot on the right there the distribution of fossils that you might find, or that we found from the literature, it doesn't look half so bad. So new ways to find fossils, something that's important about the last 25, 30 years. Obviously, serendipity is a key part of paleontology, having a good eye, knowing where to look, not aerobrading all those ectoparasites that you might be looking at. But of course, there's other imaging techniques that have come along, and I'll show you some of those in a moment. This serial grinding is simply taking a small layer of a fossil, imaging, taking a small layer of a fossil. Basically, you're destroying the fossil, but you're retaining the image. I'll look at that in a moment. Mass spectrometry for elemental analysis, which we'll have a look at also. I probably won't cover much of the molecular side. So I've spoken briefly to paleontologists, about this, and now's my chance to speak to parasitologists. Another thing is to basically increase awareness that the fossil record might be extremely useful. So from a parasitological perspective, thinking about things that might fossilise or lithify, or signals that might be retained through the past is a useful piece of information to pass on to keen paleontologists. So when looking at fossils and considering fossils, there's a few things to think about. First of all, the most important part would be to have direct evidence, to have something, an organism or a piece of an organism that's readily identifiable in place that you know is the fossil and it's interacting with something, regardless of the developmental stage. Indirect evidence is a key one, but then you're starting already to get a little bit wobbly here. Looking at telltale pathologies, swelling or boring, pits and attachment scars and so on, which we'll come to in a moment, and looking at these suggestive associations. The other part of this is that we tend to use, because we live in the present and we forget about the past, we often use what's evident to us now to make those inferences in the past. And that can work both ways. Sometimes it means we're seeing something accurately. Oh, look, it's a mosquito, it's a flea, it's a vector. It's likely a vector when it's seen as such in the past. Other times when we don't recognise the fossil form, there's no modern equivalent, or we're taking a leap of faith from taking the extant taxa backwards, it can be problematic. Here's a case in point. These are Devonian fishes, placoderms. The fish themselves are long extinct, and this group in Latvia were looking at thin sections. These are exceptionally well preserved fish, and finding hooks with or without handles. Those particular Rostella hooks, I've already given sort of the game away. Rostella would be suggestive of cestodes, but they may or may not be cestodes. But the point is that these are all over the fish, and they are potentially indicative of being platyhelminth fossils. So there's a date there that we can use later on. I talked about images, and I really do apologise for the quality of the images because these are quite spectacular. That little arrow at the top is indicating a mite that was found by Jason Dunlop and co, who did micro-CT scanning of spiders, and it just happened to be that these are fossil spiders. And actually discovering that the image on the far right is a much better image of an SEM where they could identify this critter. Again, serendipity comes in place, but suddenly you've got a fossil with a date and association. Just this week, it's not necessarily parasitic, but it shows you the extent of where paleontologies come from. This is a group involving quite a few people from the Natural History Museum, Greg Edgecombe and Jania Meyer particularly, where they've looked at Cambrian worms and discovered that they have little sucking worms attached to them. So there's a reconstruction on the top left there. And the size I put here, the size bar at the bottom, the scale bar is 400 micrometers, excuse me. So you can imagine now with exceptionally well preserved fossils, these are Chenjaean fossils, and these are soft body organisms, the opportunity possibly to start looking at finding parasites is quite exciting. And then another example, this is also serendipitous, the Civita brothers and Derek Briggs and so on, who were using these serial grinding mechanisms, were looking at a fossil ostracod and discovered, this beast in red, that they had a pentastomid attached to it. Now, they only discovered this when they analysed all the data, because it was serially ground away, it's bye-bye pentastomid. So this exists now as a set of data. But this is a key one. Pentastomids, some of you might recognise, tongue worms, only known from tetrapods these days. It was never really thought what it was only speculated as to what their intermediate host might be. This single finding of a fossil pentastomid, which is 425 million years ago, it's not only the first adult fossil that's ever been found of a pentastomid, but it's a novel host association. It's in an ostracod. It's not associated with a tetrapod, as you'd only find today as an adult. And it's ectoparasitic, as opposed to endoparasitic. So it suddenly changes things and pivots quite a number of questions when one thinks about pentastomid. I can't imagine there aren't too many people interested in pentastomid, but you can see suddenly having the right fossil at the right time can make you think about something differently. Thinking about vectors and looking at new ways to look at fossils, some work involving mosquitoes, from Eocene mosquitoes, and Ralph Harbach from the Natural History Museum was part of this to identify the mosquito. But the key part about this is the group were using spectroscopy to look at the blood-fed gut of this entombed mosquito, and we're using quite sophisticated spectroscopy to discover heme groups and porphyrin, and to recognise that this was definitely a blood meal. This is not saying there's a parasite in there, but it is saying that this is something that's sucking blood. So that's another interesting one too. But if we look more broadly and start thinking carefully about the actual groups, things like nematodes, for example, where are they found? George Poinar, for many, many years, he's the guy who inspired Jurassic Park, and Hendrik Poinar, his son, who's been trying to get DNA out of amber-preserved blood-sucking insects for many years, and failing, I have to point out. But George Poinar works extensively on amber-preserved material of all sorts of things. And you can see this insect at the top with a ton of nematodes coming out as it was dying and flailing away inside the resin. Amber is restricted in terms of where it is geographically and in terms of its ages. But that distribution there, now this is not necessarily a continuous fossil record, it's really the earliest and the latest, demonstrates that the nematode world has quite a lot of fossils out there, including some eggs from an iguanodon that Poinar managed to find through looking at coprolites. Difficult to say what those eggs were. This is part of the problem. So I've spoken about pentostomids already in the crustacean world. It's quite obvious. There are copepods out there, and then there are many crustaceans with swellings, these bopyrid swellings, which today are indicative of being isopods. Is that true from then? Can we use visualization techniques to look at that further? I don't know. But we've got these fossil horizons which are providing these new groups. Another group whizzing through isopods, and again I apologise for the images. This is work by Christine Nagler, looking again at phosphatized fish and really excellent fish specimens, but she's been looking at cymathoid isopods from this horizon and this one, and discovering not only that she can use all the morphological characters to put into a modern phylogeny, but by putting it into a modern phylogeny can demonstrate that the age of divergence of that group must have been considerably before the age of this particular fossil. So it's a real age on fossils. So I work a lot on platyhelminths and trematodes, and this is a phylogeny on the left, a relatively crude one that we have at the moment that's as good as we've got, and then a list of the first intermediate hosts, second intermediate hosts, and definitive hosts as cartoons on the right. And I've circled that little snail there. In fact, it shouldn't really be a snail. In this case, it should be a bivalve. About the only thing we can think of in terms of finding a fossil trematode are actually gymnophallid trematodes. These are things that cause little blisters in bivalves today and little pits or igloos, and they're quite characteristic. You can find them today, but you can also find them deep into the fossil record. Now, the one thing about these trematode-induced malformations, and this is Kenneth's thing, he looks at pearls in ammonites, and he also looks at pits in bivalves, is that, as he says himself, that distribution of parasite pathologies that he sees on the far right of the image really doesn't tell the whole picture, because in his assessment, where those particular fossil bivalves have come from suggests biased sampling, poor taphonomy, the process of fossilization, and incomplete sampling. But he's one of these people who will go through museum drawers now to look for additional evidence. So it might be that we can actually begin to push back and get a better accurate date for at least one node in the trematode tree. I've told you I work on cestodes. This is a tree that we produced on cestode phylogeny with an indication of the host groups, and you can see a lot of elasmobranchs across the radiation of cestodes. They're pivotal to the radiation of cestodes. The only fossil cestode that we have beyond the archaeological side, but quite bizarrely and quite amazingly, is a 270-year-old, million-year-old coprolite within which that was thin-sectioned again. Not only can you find eggs, but you can find the eggs laid out as a proglottid. What they were, and more disturbingly what the shark was, we have no idea, and that's a problem there. So if we took a time tree and we took this calibrated phylogeny and we said, okay, we have an indication of the origins of life, and throw the soft body things in, we use the same molecular clocks that everything else has, we can come up with a varied set of dates of what might have happened in terms of the major radiations of both the platyhelminths, which are starred here, and the parasitic phylums. But you can see the range of dates between 403 and 600 million years ago from a time tree. So one of the things that we're trying to do is to see if we can make that better. And again, apologies for the image here, but that list of characters on the far right is basically a set of new fossil dates for the platyhelminths that we've got, some direct, some indirect, but we think we might be able to calibrate this tree a little bit better. So a lot of you work on poop, and a great source of fossil parasites would also be coprolites, and just a listing of what's been found in coprolites here in terms of the diversity of animal phyla and other parasitic taxa, not pollen being parasitic, I appreciate, but things that are of a similar, of a size that might be able to be used to indicate fossil worms is here. Again, if we think about the body fossils, the actual bodies of fossil parasites and some vectors in this case, that list on the far right tells you what's actually been found before, so it's quite a diversity. There's some key groups missing. Mysostomids, orthonectids, acanthocephalans are poorly represented. So they, again, is it because they're not there, or is it because we haven't looked for it? And I just wanted to remind you here that the kind of things that are going to get fossilized in the first place are likely from these kinds of deposits, almost in this order. So amber, great, coprolites likely, things from freshwater and marine systems, those deposits in caves, but not really terrestrial material. So as a quick summary, before David leaps and takes me off the board and I present him with his P45 as a gift, I'm his head of department. The quick summary of role of fossils in parasitology. So there's what does it mean and what can we do with it? We can get confirmation of particular associations. We can talk about the paleoenvironment, things I haven't mentioned in here. We've got those real age calibration points that we can throw in. We can look at the nature of associations, for example, that pentastomid example. We can look at developmental stages, again, the pentastomid being an adult, not another fossil larva. Parasite features, the attachments, and so on and so forth. And then looking at the techniques, the ancient DNA, proteomics, elemental analysis we haven't gone into, but that's another area where we could be targeting better understanding of elemental analysis of lithifiable material today could be identified in fossils later on. We've looked at CT and micro CT, but CT is getting better and better all the time, being able to differentiate the smallest differences in densities. We know about this from soft tissues, but it also works for lithified material. And then, of course, there are always caveats in terms of interpreting any of this stuff. So in absolute summary, direct evidence in the fossil record is rare. Modern imaging techniques are revealing increasingly more examples, even in unexceptional fossils. Elemental analysis is yet to be fully explored. And I thought in the theme of today, those of us talking about different kinds of parasitism and getting rid of it, so parasite elimination is difficult, we recognize. Eradication may seem impossible, but at least in a geological sense, extinction is inevitable. And that's it from me.